Thanks, Karen. So, as John Cleese would say, now for something completely different. So, I'm drilling down in this talk on what the actual mechanism of the NovoTTF-100A is. So, this is a result of a sponsored research agreement that we've had with Novocure. Let's see if I'm challenged as well. I am. Okay. So, yeah, I don't know where the disclosure slide went. I just gave the disclosure. So in terms of how electric fields can potentially affect treatment within patients, and if you think about it, this is going to naturally get a lot of pushback from people due to all of the electrical devices that were proposed and used in the early part of the 20th century. If you actually think about how this might affect treatment in a real sense, you have to think about what the charges are going to act on. So I'm by training as much a cell biologist as anything else. I do have an immunological background, but it's through my cell biology training. So I think of everything in the context of what's going to go on within the cell. And so if you apply, if you just think about it, I will get this down. So if you think about it, we all know I'm gonna have to be trained before I come up here. So if you think about it, everyone knows that opposite charges will attract each other, and anyone who's run an electrophoretic gel knows that if you've got a charged molecule and you put it in a constant field with a positive and negative pole, negatively charged molecule will go towards the positively charged pole. The NovoTTF-field is not a constant field, but an alternating one. And so you have the field in kind of a square wave alternate between positive and negative between the two poles. And so if you've got a simple negatively charged molecule, it's probably just going to vibrate there. However, if you've got a molecule where it's got dipole moment, where the charges are separated from each other physically, such a field could potentially cause rotational or torsional stress by first, flipping this side of the pole up and then pulling it down. And so you can imagine that if you've got the right dipole configuration, the molecule within the cell should rotate within the, based on the presence of the field and based on the frequency at which it actually runs. So as many people in the room know, the NovoTTF-field system, the NovoTTF-100A, has been FDA approved for recurrent glioblastoma, and this is based on the EF11 phase 3 trial that showed equivalent efficacy to standard of care plus an increase in quality of life. And it was mainly the quality of life factor that got the approval. So this is a portable home use medical device. It's got a non-invasive disposable transducer array that's orthogonally so you got basically two arrays. They're orthogonally placed, and so these provide alternative. These provide the alternating field from this pole to this pole and from this pole to this pole. So you get an intersection between the poles at most points. So it delivers the TTFields therapy continuously to the tumor within the brain, and the important thing here is that it's a very narrow range at which the TTFields operate therapeutically and in the dish. And this is between 150 to 200 kilohertz. If you have significantly higher or lower frequency, the TTField doesn't work. So this is the original movie that Dr. Joran Palti made. He did this in his backyard in a shed that he set up. So it's having done a lot of these long-term cell biology movies, it's amazing that he was able to get this to work with such a primitive setup. But could I get this movie to play? Thanks. So the thing that I want you to focus on is that when cells round, and he circles these, and goes into mitosis, you see this massive blebbing. So they interpret this as apoptosis. And when I was looking at this movie more closely, it was very apparent this is not apoptosis. Can we see this play again? So what I want you to see, what I want you to watch is the fate of the cells after they've blebbed. So you can see that. Those are the first one, second one. Goes into blebbing, and now look. See them? So once a cell undergoes apoptotic blebbing, it's dead. There's no recourse. That's a row A cleavage event, and the caspases have gone to shear the DNA by that point and everything else. And all these cells that are in this movie that were blebbing, they all survived. They blebbed for a while, they stopped blebbing, cells crawled out. So it became obvious to me that this was not actually a process that was cytotoxic. It was something else. And so just a little primer on the cell cycle. You go through interphase, which is what most of the time for the cell, for the cell's life, they're in interphase, and then they go through mitosis, and you've got prophase, prometaphase, metaphase, anaphase, telophase, and then you get cytokinesis in 2 cells. So this will become important in the next few slides. So, in terms of phasing out exactly where the NovoTTF affects the cell cycle, there are certain events that occur that are dependent on other events. And the entry into anaphase is critically dependent on, first, the capture of microtubules by the kinetochores of condensed chromosomes at the mitotic plate. And this capture causes the activation of APC, which then will cause the destruction of securin, which normally blocks separase, and then you get into anaphase. So therefore, the entry into anaphase is prima facie evidence of healthy microtubules. So the first thing we looked at is we looked at markers for mitosis and comparing cells going through, synchronized cells that were going through M phase that were either sham treated or TTFields treated. We didn't see much of a difference. Just like the movies predicted, cells transit through these parts of the cell cycle. And compared to Taxol, which causes metaphase arrest, you can clearly see that these cells are relatively unperturbed. But if you look more closely, you find, just as NovoCure had already demonstrated, you find that there is a defect in mitotic progression that's more subtle. They spend more time in the cell cycle. So we wanted to pin down exactly what part of the cell cycle was prolonged. And so we did Western blots looking at targets of APC. And so we find that if we look at securin, which is a primary target of APC, we find that there's really very little difference between the TTField treated and the control. And again, you see the paclitaxel has a major problem getting through this phase of the cell cycle. So if you look at phosphohistone H2, on the other hand, which remains phosphorylated until the end of cytokinesis, phosphohistone H2 is clearly elevated for a longer time. So these two data together strongly indicated the problem is in anaphase. The problem is not in metaphase proper. So we went and did a long series of time-lapse observations. And could I get the top movies to play? So what we did was we synchronized cells again, and then we watched them. Yeah, we watched them divide. And up on the top, you see sham treatment, and you can clearly watch the cells undergo division. If you can play this again. So you see a mitotic plate, and then you see division. Whereas what you see in TTField treated cells when you watch the this is the metaphase plate. This is stained with DRAC5 in order to visualize the chromosomes. So you can see the metaphase plate, but as soon as you see the cell go into blebbing, you obviously see this perturbation of the standard metaphase plate. I've taken individual frames here, and you can see that this perturbation of the metaphase plate is coincident with this blebbing that we saw in the first Urim-Palti movie. So we looked at the time intervals between rounding and metaphase plate formation and metaphase plate formation and the exit of mitosis, either through blebbing or through division. And we found that in both cases the times were identical. So there's no difference between the ability of the cells to produce a metaphase plate and the ability of the cells to then go on and either divide or bleb. The time intervals are the same. And we see no interval, pre-anaphase, we see no problems with the formation of mitotic spindles, but after anaphase, we see all these disrupted cells. So this clearly showed that these cells were initiating this blebbing that Urim-Palti originally saw at the metaphase to anaphase transition. And so here you see that the consequence of the consequence on the DNA is, by studying the DNA, you can see the consequences, the preservation of a large number of 4N cells, so these are cells with double the amount of DNA that daughter cell should have. So many of these cells have just simply tried to divide and gave up. And then they crawl out and they become these 4N cells. So that means that the TTField, unlike traditional chemotherapy agents, is actually acting at a very late part of the cell cycle. And at this part, there really isn't an evolved mechanism to prevent the cell from becoming deranged in the post-mitotic state. And we additionally see evidence of this in MCF7 cells, it's a peculiar cell line. They become very, very large when they become tetraploided. And again, we see a large number of these very large cells in the TTFields treated case, but not in the control. So what's the target of the TTFields? And this is the main point. So we looked at the criteria for which the criteria for the target of the TTFields, and we figured it's got to be a protein that has a high dipole moment, obviously. The candidate has got to be involved in a biological process that is perturbed by the TTFields, and what we've determined in these previous experiments is that the TTFields cause ectopic furrow production. So these are what's referred to commonly as mitotic blebs, not apoptotic blebs. And so the TTFields should be established the TTField target should be essential for establishing or maintaining a structural order that the input energy from the field is able to disrupt, and the TTFields are not likely to significantly perturb the function of proteins that are in high entropy states, such as those that are free floating or in the in membranes or in the cytoplasm. So they have to be in some type of stable structure. So I looked through all the a large number of candidates, and I came across a protein complex called Septin, which is the fourth cytoskeletal element. So the Septin heterotrimer that's made up of these Septin family members, Septin 2, 6, and 7, has an extremely high dipole moment of 2,711. Tubulin, which had previously been thought of as the potential target, is way down here at 1,600. And so that's 2 standard deviations from the norm. This is 5 standard deviations from the norm. And interestingly, if you knock down Septin knockdowns as they try to go through mitosis have the same phenotype as the phenomena you see with the TTFields. And the TTFields TTField treated cells become larger. Septin depleted cells also become larger. So if you look at Septin, Septin localization in TTField treated cells, normally Septin is localized at the midline. You can see it here at the cleavage furrow and at the Anaphase spindle. But in TTFields treated cell, this localization is completely disrupted. Interestingly, at 500 kilohertz, which is not a killing frequency, these you only get a partial disruption. And we quantified this, and we actually show that this is a significant decrease in localization, but it's not as much of a decrease in localization as you see with the killing field. So this correlates nicely with the ability of TTFields to kill cells. And we stain for pole like kinase 1, which stains the midline. We found there's nothing wrong with the midline itself. It's just a failure of Septin to localize to it. We looked at another Septin localization phenomena, which is localization to the mitotic cytoskeleton upon spreading. And we also found an extremely similar effect on Septin in this localization effect. So it's probably not some upstream event that's specific to mitosis. And like I said, Septin knockdown cells are larger than their parental counterparts, and this is also true for TTField treated cells. So we're pretty convinced at this point that at least one of the major molecular targets of the TTField is actually Septin, in the Septin. And the Septin cytoskeleton actually demarcates the outer edge of the furrow, and it sets up like a large picket fence that is, the ultrastructure is like a large picket fence that goes around both sides. On the inner side of that fence, you can get contraction. On the outer side of the fence, you can't get contraction. And this is why Septin knockdown and potentially the TTFields cause ectopic furrow formation. So this is kind of the model. The model is that septins have this high dipole moment. The TTField perturbs their ability to align, and this perturbs their localization, and then you get these perturbed cells as the cell perturbation as the cells try to go through mitosis. Cells that emerge on the other side have extremely high vacuolarity. They have cell cycle arrest properties similar to doxorubicin that you can see through ATP, shift in ATP metabolism. They have decreased proliferation if you treat cells within the field, if during M phase but not in G phase. And you can see that there's decreased BRDU incorporation in P53 positive cells, but it's much diminished in the P53 negative cells. And likewise, if you look at apoptosis downstream, apoptosis does not happen within the field. Apoptosis happens subsequent to the field. So you get a peak of apoptosis at about 36 hours, and this apoptosis is also P53 dependent. Now, cells that are experimentally tetraploided like this will undergo immunogenic cell death. So Cromer showed this a few years ago. And so we checked this out, and sure enough, we have the hallmarks of immunogenic cell death within treated cells. We have calreticulin upregulation, which is a kill me signal, and we've got HMGB1 depletion, similar to what you see with doxorubicin. So this calreticulin upregulation is also similar to what we see in terms of septin localization. At 500 kilohertz, you see an intermediate level. And the idea that this is an immunological approach will be dealt with more when Eric talks. But there was an original piece of research that they did at Novocure where they did a VX2 tumor injection into the kidney capsule. And this is a highly metastatic tumor. And it goes to the lungs, primarily. And if you treat the tumor for 7 days with TTFields and then let the tumor go for a month and a half, and then look at the level of metastasis, prior TTField treatment lowers metastasis, and the Mets that are present in the lung have an increased CD45 infiltration, which is probably protective. So this is the model for how the therapy is likely to actually work in patients. You have TTField treatment of mitotic cells. These mitotic cells exit mitosis aberrantly. You get ATP, HMGB1 secretion, and calreticulin upregulation. And then that attracts in and activates immune effector cells, which then results in tumor regression. So I'd like to thank all the people who helped me on this. I'd like to thank Eric for providing for providing the support that I needed. And Talia and Aaron and Sessian did a lot of the bench work with me. Bert Vogelstein gave me the HCT 116 P53 plus and minus pairs. Nidhi at Harvard Medical School did all the time lapse with me, and Moshe Galati at Novocure was indispensable with all the help that I needed to get the TTField treatment devices in vitro up and running. Thank you.